Today we're going to be talking with Erica Newman. She is a PhD candidate in the Energy and Resources Group at the University of California, Berkeley. She studies wild bird and plant communities, natural disturbance regimes, and how anthropogenic change interacts with all of these. Her research spans multiple systems, including high Sierra Nevada meadows, Bishop pine forests, and California chaparral. Case examples of how natural disturbance and anthropogenic change affect community structure metrics such as species diversity, rarity, and the interactions among affected species. And prior to her work here at UC Berkeley, Erica received a master's degree in physics from the University of Michigan, and then went on to work in bird ecology and prescribed management in the state of North Carolina and the National Park Service. So our webinar, a few more housekeeping things today before I hand it over to Erica. This webinar is being recorded, and once the link is posted online, I will send an email to everyone who is registered. It can be found on our website, California Fire Science Consortium, and that is going to be linked at the end of the presentation as well. I will also mention that all of the names, everybody's introducing themselves, I'll make sure to remove any of those identifying factors before we post it online. As we're going through this presentation, you can see I see people are making great use of the chat box to introduce themselves. We're also going to use that chat box to record questions. So throughout the presentation, you can type a question there, and I'll collect them, and we'll have a Q&A session at the end. Additionally, you can save them for the end and paste them in once we get there. So I think with that, that's everything from me, and I'll turn it over to Erica.
Okay, thank you so much. Thank you everyone for joining me today for your lunch break. I am pleased to be here. I wanted to thank Stacy so much for organizing this. There's a huge amount of work that goes into organizing a webinar. And I wanted to thank Stacy and Liz Van Mancham for inviting me to give this webinar. So today I'm going to talk about fuel management in Chaparral, its effects on wild birds in California, and how mastication is changing bird communities and maybe increasing Lyme disease incidence across the state. Let's see. Sorry, I gotta get used to the controllers here. Okay, so first I'm going to talk about, I'm going to give you a little bit of background about Chaparral in California, and then I'm going to talk about two separate studies that I'm doing that are linked. The first one is a Lyme disease study that was done in the lab of Robert Lane at UC Berkeley. And the purpose of the study was to figure out which species of birds are competent hosts of the bacteria that causes Lyme disease in humans. The second study, which is related, is a comparative effect study of fire and mastication in different seasons in Chaparral. And these are linked because they're both done at the same field station, and they both overlap in the Chaparral. So at the end, I'll give a synthesis of what's going on with landscape change, bird communities, and Lyme disease. Okay, so the research study area that we worked at is the Hopland Research and Extension Center that's about 100 miles northwest of San Francisco. It's in the Mayacamas Mountains in the North Coast Range, and it has multiple different ecosystem types. For the fuels management study, we worked only in chamise-type chaparral, which had large proportions of ceanothus and manzanita in it. So here, let me give you just a range map of chaparral in the state. There are 7 types of chaparral in the state. In Southern California, it's much more diverse. In Northern California, you get different types of chaparral, and it's much less clustered. It's already sort of a patchy distribution. The chaparral is part of the California floristic province that goes from southern Oregon to northern Baja California. And the thing that you need to understand about the California floristic province is that it's basically the only biodiversity hotspot in North America. It contains about 3,800 plant species, half of which are endemic to the California floristic province, 20% of which are endangered. It has about 1,500 native bee species, And it has the largest, it has the most bee diversity per area in the entire world. And a lot of this diversity is in Chaparral. Chaparral only covers 6% of the state, but it kind of carries a big share of the biodiversity in the state. So it's worth studying, and it's just beginning to be protected. Some national monuments are being put in Chaparral now, but it's generally not protected as a plant community. This is important to note because there have been a couple of studies recently that show that with climate change, we're going to be experiencing more fire, probably in Northern California under hotter, drier scenarios. This was a study that was done by Enrique Batlori and Max Moritz and others, published in Global Ecology and Biogeography in 2013. Other work has shown other works by Volker Radloff has shown that there are about 5 million housing units in the California floristic province, specifically in the wildland-urban interface. And pressures on the chaparral are only likely to increase. So a study by Michael Mann, Max Moritz, and others have shown that probably about 500,000 more housing units will go into the wildland-urban interface in the next 35 years. So that's just some background on why it's important to think about protections for chaparral. Okay, so the first study that I'm going to talk about that was done with Robert Lane and Lars and Rebecca Eisen, who are at the Centers for Disease Control and Protection, Jayammoon Hasty, who's in Hawaii, and Natalia Fedorova, who did all of the lab work for this. I'll talk about this first. I'll note that Charles Vaughan did all of the bird identification on both of these projects and is just a wonderful resource. So what is Lyme borreliosis? Lyme borreliosis is infection with a bacteria, a spirochete bacteria called Borrelia burgdorferi. It's generally carried by ticks. In the West, the tick that carries it is the black-legged tick, Ixodes pacificus. In humans, Lyme borreliosis is called Lyme disease. It's North America's top emerging vector-borne disease, with 300,000 new cases being diagnosed or estimated annually in the US alone, although Lyme disease actually has a global distribution in temperate regions. So some cases of Lyme disease are quite mild and can be cured, but other Lyme disease cases are chronic. They cause arthritis, fever, headaches, rashes, fatigue, depression, cognitive impairment, and memory loss. It's a very serious disease, and it's also been linked to autism in developing fetuses. So it's very important to understand why Lyme disease is emerging and why it's becoming more prevalent in the United States, and where it's coming from. It's definitely present in California. It's not as big a concern in California as it is on the East Coast. So on the East Coast, you have about 90 to 95% of all cases in the United States. Lyme disease is present in the Midwest and the southeastern states. In California, you have some hotspots of Lyme disease, but really much lower incidence. But something interesting to note is that after I published this study, which I should note is in PLOS One, and it's free, open access, available to everybody, including managers who want to look at the details of this. I was contacted by a number of people who said they had Lyme disease, they contracted it in California, and it went for years untreated because their doctors didn't think that Lyme disease was present in California. So one of the things that we need to do is actually raise awareness that Lyme disease is present in California. And it's probably very underdiagnosed in California. Okay, so you note that Hopland is actually in one of these sort of hotter spots of Lyme disease outbreaks. Let me give you some definitions. Lyme disease is an emerging infectious disease. That's a disease that has increased rapidly over the increased in incidence rapidly over the last 20 years and is likely to increase more in the near future. Like Lyme disease, emerging infectious diseases often arise from ecological change. In fact, Lyme disease is the example that people often give, although Ebola is another good example of diseases that come out of wildlife and result from ecological change to increase in incidence in the human population. 60% of emerging infectious diseases are zoonotic in nature. Zoonoses are infectious diseases that are transmitted between non-human vertebrate species like birds and mice and rats and so on to humans and possibly back. I'm going to give you just a brief taxonomy of this spirochete bacteria that causes Lyme disease. So Borrelia burgdorferi sensulato means Borrelia burgdorferi in the broad sense. And it's a group of closely related organisms that are known to cause Lyme borreliosis. So there are at least 6 of these species in California. At least 12 of the species overall cause Lyme disease-like illnesses. So Borrelia burgdorferi sensu stricto, in the strict sense, is BBSS. This is the prime cause of Lyme disease in the United States, and that's a big focus for us. And Borrelia bizzettii is an interesting disease that causes a Lyme disease-like illness in some people in Europe and northwestern California. But that one is less studied and less well known. So in the BBSL grouping, we're going to be focused on BBSS, cause of Lyme disease, and BBIS, the other interesting disease here. So unlike the East Coast, where you have deer as one of the prime resources for ticks to increase in number, the story on the West Coast is much more complicated. The vector that we have, Ixodes pacificus, interacts with the pathogen, and the pathogen, BBSS or BBSL, is often carried by a host, and these hosts may be the dusky-footed wood rat or the western gray squirrel, which can contain the pathogen in their blood and don't get rid of it. We also have southern alligator lizards, and we have western fence lizards that also get bitten by ticks. And you can see in this picture ticks around the ear hole right behind the mouth of this southern alligator lizard. These animals have been shown to clear the infection from their blood. So they may be actually sort of lowering the overall incidence of Lyme disease bacteria in wildlife populations. And that's not a dynamic that happens on the East Coast. So in this study, we're asking about birds. We have about 400 regularly occurring species of birds in California. And we want to know, are they hosts for the vector, or can they actually competently carry the pathogen in their blood? These cute fellows are the ticks. The one in the bottom right corner has fed up on blood. Kind of disgusting. I tried to pick pictures that weren't terribly gross. But as you can see, let me see if I can do this. I'm trying to draw here. Yes, here we go. Okay, so the life cycle of the tick, the eggs are laid without infection. And therefore the larvae here are completely without infection because they haven't fed on anything yet. When a larvae feeds on a primary reservoir, like this rat here, it can become an infected nymph. And then the infected nymph will interact with incidental hosts, which it may also infect. So it can infect humans. Turns out that deer are not competent carriers of the Lyme disease bacteria, but they do feed the ticks and increase the abundance of ticks. And then we have adults that are not sort of interacting with the wildlife hosts. Okay, so let me turn off this drawing thing. Okay, here we go. So what about birds? Birds definitely overlap for the active part of the year with ticks in their range. But based on their behaviors, they may or may not come into contact with ticks. So we want to come up with a way of understanding which birds are infected, which birds can transmit the infection to other ticks, and which birds are really interacting with ticks in a way that would cause them to come into contact with this with infected black-legged ticks. So so this is how the tick works. The nymph will climb up onto a grass stalk or something low to about a meter in height or less, and it uses its Haller's organ to detect hosts. Now, I wanted to just say here that when I think about ticks interacting with birds, this is a very, very old interaction. There's a book called What Bugged the Dinosaurs? Insects, Disease, and Death in the Cretaceous by George and Roberta Poinar. And it makes the case that dinosaurs really interacted with ticks because ticks have been around for about 200 million years. Dinosaurs, over 60 million years. So birds now are the only living descendants of dinosaurs, and taxonomically that actually makes them dinosaurs. So this interaction between ticks and possibly between spirochetes and birds has been going on for a really long time. So we want to understand more about the behaviors of birds, more about whether the ticks are actually attracted to the birds. Do the birds have defenses against the ticks? They could produce chemical compounds, and it's been shown that that many birds produce chemical compounds that actually paralyze and kill lice. But similar studies have not been done with ticks. Okay, so we have all of this going on in this system, and we want to establish wildlife disease reservoirs. So we know to do so, we use these facts. A bird can get spirochetes only from an infected nymph. The spirochetes must survive in the reservoir bird, and the reservoir bird must transmit the infection back to a different tick. And so here we're only considering the transmission to larvae from an infected bird because the larvae are infection-free before they attach to a host. So we're interested both in the number of ticks in birds, or infestation, and infection, spirochetes in the blood or in the larvae. This is how we collect the data. Ticks tend to group up near the head and eyes of a bird where they can't remove them with their own beaks. So we put up nets. These nets are sort of like badminton nets, but they're very, very transparent, and the birds will fly into them. You catch the birds, you remove the ticks and put the ticks in alcohol. You send those off to the lab, and then you release the bird. You mark the bird by clipping a tail feather so that you don't recatch the same individual. And then you have data. So we had data from 14 sites at Hopland. Nets were open 4 to 8 hours after sunrise. And total sampling time in the nets was over 3,000 hours. We caught 623 birds with 284 ticks. 23 of the 53 bird species that we caught had Borrelia infection of some sort. 100 of the individual birds carried ticks. And let me show you the results from the blood analysis. First of all, you notice, let me see, let me do this drawing thing again. You notice that birds, oops, sorry, birds have primarily BBIS in their blood, this disease that's less well known than BBSS. So this study was the first study to ever discover BBIS in birds. So that's very interesting because it wasn't known that birds were even part of the BBIS disease ecology story. But when you look at larvae here, 96% of the infected larvae carried BBSS. What this means is that birds are preferentially or disproportionately, let's say, passing on BBSS out of the infections that are in their blood. This is very interesting because this is an amplification factor that comes from birds. Okay. We also want to know generally what predicts whether a bird is going to be infected and whether a bird is going to come into contact with larvae or nymphs. So this will allow us to move beyond what's been done in Lyme disease before, which is just sort of assessing the number of ticks that are on birds and looking at numbers from site to site. We want a model that tells us where are these birds getting infection from? What explains why they're infected, or what explains why they're not infected? So we look at presence of infection and infestation, and then we also created zero-inflated negative binomial models that are counts of larvae, that predict counts of larvae and nymphs. So I included a number of explanatory variables to explore, including the year that the study was done, because it was done over the course of 2 years, body weight of bird by species, number of nymphs removed from a bird for modeling infection, and main habitat type. And let me just say a word about those main habitat types. We looked at 4 main habitat types in this study. From left to right, these are oak woodlands, which is sort of an open canopy oak savanna with grass understory. Dense oak woodlands, which has very little understory and is mostly covered, has a mostly covered canopy of oaks. We looked at grasslands. This is a picture of naturalists in Hopland exposing themselves to ticks. And we also looked at Chaparral. So this is a picture from the Northern California Prescribed Fire Council visiting the sites that the next study was done at. And just for scale, you can see that the chaparral comes up to about people's chests. Okay. We included a number of variables that relate to bird behavior because when you look at these different birds, you see that California quail, which walk on the ground, they forage on the ground, and they nest on the ground. The lower picture is California quail chicks, are going to be very different in terms of their exposure to something like a bush tit, which is a foliage gleaner, spends most of its time in bushes and trees and up off of the ground where the ticks are, and even makes its nest high up in bushes and trees. So it's not going to come in contact with ticks quite as much. And it should be just pretty clear that what you eat and where you eat it is going to affect your exposure to ticks. So we include things about what the birds are eating. Are you a nectarivore, like this Anna's hummingbird? Do you eat birds, like this American kestrel? Are you a frugivore, like this cedar waxwing? Or are you eating something else and nesting somewhere else, like this acorn woodpecker? Okay. So these are the explanatory variables. We looked at more explanatory variables: main food, foraging substrate, nesting substrate, and the presence of the bird, whether they migrate or not, and whether they're in breeding condition or not. And we're just sort of asking whether these categories of variables explain anything, and if so, which particular behavior is correlated to infection and infestation. We also added a component that was new for this study and not done in the field yet, by which I mean Lyme disease has not incorporated taxonomic predictor variables related to the relatedness of hosts. So here we have 2 orders. We have woodpeckers in one order and songbirds in another order. We have 2 families of songbirds. So we had quite a number of families of birds in these two orders, woodpeckers and songbirds. We want to know if taxonomy is actually more important than behavior, because, like I said, bird immune systems and bird odors and bird chemical defenses may actually have a role that has evolved over evolutionary time to prevent them from being infected or even infested by ticks. If we look at the data, we can see just the raw data that larvae are just sort of out of control in the grasslands, and nymphs are more present in the grasslands, but we really need a model to tell us what the significance of these variables are. So here's the results from the statistical modeling. What predicts infection incidence in birds? The first thing was year. So moister years have more ticks and larvae. The number of nymphs removed from the bird was positively correlated to the infection in the bird, and order. So we basically see that BBSL as a group is like the group of bacteria that are going to be involved in Lyme disease and Borrelia bizzettii are going to be sort of confined within songbirds. And the other order that we were looking at, woodpeckers, were just not involved. If they had ticks at all, they weren't infected. And this could be quite general. And if we had more data, we might be able to say more about whether we really should only be studying songbirds, because those are the birds that are involved in the maintenance of the bacteria in the wildlife population. What predicts the number of larvae on birds? In descending order of significance, grasslands, like I showed you, was really out there. Oak woodlands also had a very high number of larvae per bird, and dense oak woodlands. What you don't see here is chaparral. Okay, so what predicts the number of nymphs on birds? Again, in descending order, being a bird that's associated with grassland habitats, being a bird that's associated with oak woodland, and finally being a bird that's associated with dense oak woodland. Number of nymphs is also predicted by feeding substrate, although none of the levels within feeding substrate seemed particularly significant. In the presence only things, we can see that ground and bark foraging birds are more highly exposed. But in terms of the number of nymphs on the birds, no particular level stood out. Okay, so what do we have? We have chaparral as this kind of thing that didn't show up as being positively correlated with infestation. We know that chaparral is full of ticks, but it's significantly negatively correlated with infestation in both cases, both for larvae and for nymphs. So to be clear about that, it's not that chaparral doesn't have ticks. It's that the birds that primarily use chaparral as their habitat, like California thrashers, like wren tits, they are not carrying infestation of ticks. Other major findings that we had from this study, we had 3 carrier species that we could identify that were passing Borrelia burgdorferi, the one that causes Lyme disease, back into the wildlife population. These are American robin and dark-eyed junco, which were known from other studies to be carriers. And we also identified the golden crowned sparrow as one of the species to watch. Juncos and robins have also been noted from another study to increase in abundance with suburbanization. This is a study that also came out of UC Berkeley from the Rory Bowie lab. Chaparral-associated birds carry blood infection, but it's mostly Borrelia bizzettii, which is kind of interesting because it means that the kind of where people are picking up bizzettii and why is probably a more complex story than that we don't understand yet, that we may be able to get some handle on through the bird community. And finally, bird blood is not the tissue where BBSL resides. We had birds passing on other species of Borrelia than were detected in their blood. This means that we may be really underestimating the role of birds in the maintenance of Borrelia burgdorferi and Borrelia BBSL in the wildlife community. Okay, so that's that study. What happens to chaparral-associated birds under management scenarios? The answer to that is going to be in part determined by another study that I'm working on with Scott Stevens at UC Berkeley. Bird community response to fuels management in Northern California chaparral, effects of treatment in season. Again, I'll point out Chuck Bond, who's great with birds, James Dawson, who led a lot of the prescribed fire and mastication that happened on BLM land, and Jen Potts, who collected all of the bird data for this study over the course of many, many years. Okay, so chaparral management. We want to know what happens with prescribed fire and mastication in multiple seasons. So it's pretty clear that chaparral is very fire prone. People are looking at different ways of managing chaparral to lower fire risk where fire risk is high near human habitation or comes into conflict with people. Mastication is expensive. You can't do it in remote places, and it's limited to low slope areas. Prescribed fire as a fuels management tool has the risk of escape. People don't like fire around them. There are also air quality concerns and even smoke concerns, because it seems like chaparral actually responds to the presence of smoke by regenerating its rare annuals underneath the mature plants. And this might mean that we might be exhausting the seed bank of chaparral by exposing it to smoke through prescribed fire. So I'm not going to talk about that today, but this is a great way of understanding how anthropogenic wildfire that caused by carelessness or arson is going to be affecting these communities. So following treatment, we had about 95% removal of the canopy cover. Fire was carried out in winter, spring, and fall in 2002 and 2003. Mastication was carried out in the spring and fall. In the winter, apparently, it was too muddy to do this. Treatments were implemented on 1 acre plots with 4 replicates each. This is a total of 20 treatment acres that look like these bare patches and 4 control plot acres. And the post-treatment fuel bed, or ground, if you will, fire still maintains some of the structure of the original plants, whereas mastication produces this kind of small, still flammable material that's left on the ground. After treatment, after 3 years post-treatment, we have the return of some shrub cover, more shrub cover, 71%, than in the masticated plots, where only 43% work. 43% of shrub cover returned. So that's a pretty significant difference. Work by Jen Potts and Scott Stevens and others has also shown that basically mastication has the highest number of non-natives in the plant community and 34% more non-native annual grasses by cover. Winter and spring fire have the lowest number of non-native species. So as you can see in the bottom here, the effects of chaparral management on birds is going to be tied very closely to how much vegetation cover there is and what plants are there. But I'm going to be just looking at the birds. So here is the number of individuals rather than cover of those non-native annual grasses. And you can see that there's a really stark difference here. This is 3 years following treatment. Kate Wilkin is a graduate student at UC Berkeley who has a paper in review right now that's basically showing that effect of the non-native grasses persists a decade and more after treatment. So we're really introducing kind of a fuel type and a species component that was not there before with mastication. And this is Jen Potts, Kate Wilkin, and myself at the Northern California Prescribed Fire Council talking about these studies. So what did Jen measure? This is really incredible. She did so many visits to all of these sites. With the control plots, you find that the dominant species, so these are just the most abundant species that I'm listing. There were 39 species in control on 122 visits. There were wren tits, western scrub jays, lesser goldfinches, and spotted towhees. And with fire, you see the same dominant species, and mastication, basically the same dominant species, although you have a bump up in nectarivores, possibly because you have more flowering plants. These are the dominant species shown on the right here. This result agrees exactly with a study done by William O. Wirtz in the late 1970s in Southern California, Chaparral, that basically showed that following wildfire, the community structure is unaffected. It's the abundances and the rare species that are affected. To account for unequal survey effort, we do something called species accumulation curves. This is a way of estimating how many species we would have really measured if we could have done equal sampling between sites. And so you get this flattening of the curve where more samples are not going to help you pick up more species. And I took those chow means and I plotted them here to show that basically even accounting for bias introduced by unequal sampling, control plots always have significantly more bird diversity than anything else, although fire seems to be close in the first couple of years following treatment. Mastication is really low. So what you're seeing here is the lower limit of bird diversity as limited by the number of species that we actually measured. One standard deviation up showing significance, and a 95% confidence interval on top of that. In terms of the structure of the community, you have diversity in these solid lines, and you have evenness in the dashed lines. And so diversity, Shannon diversity is just a measure of overall diversity. Evenness is sort of like if you had a room full of 100 cats and 100 dogs, that would be even in terms of species numbers. So evenness would be high. And if you had 199 cats and 1 dog, it would be very uneven. But like that situation where you're imagining all these animals in the room, it's actually evenness, or I should say unevenness, has been correlated to stability of the ecosystem. So it's more stable to have, say, 199 cats and 1 chihuahua than it is to have 100 cats and 100 dogs in a room. So the control plots have high diversity, low evenness. And you see that basically like a year following treatment, fall fire basically has that exact same pattern, whereas mastication has high evenness and low diversity. For spring, it's exactly the same. High diversity, low evenness for the fire treatments. Mastication has the opposite pattern. And winter, where we could only do fire, same thing again. And my prediction, if we could have done it, would be that mastication would show high evenness and low diversity. So that's just the community structure. I don't have a graph for this, but to give you a sense of what mastication does to the community, we have 39 species total, birds, represented in the control plots. And basically, you're left with about half of that in the masticated plots, even 3-4 years following treatment. So we don't have enough data to say this with high significance, but basically the birds that make it through contain two of the bird species that are known to be really important carriers of Lyme disease in sort of generally, but in California specifically. So these are the American robin and the dark-eyed junco that are surrounded by red here. And like I said, these by another study that was done by Rory Bowie, Morgan Tingley, and others, we know that these are birds that persist and expand and increase in abundance in suburban settings. Okay, so the other thing to be aware of in Chaparral is that rare birds basically get knocked out altogether. This is the bell sparrow. The bell sparrow is Artemiospiza belli. It used to be Amphispiza belli. And when I started this project, this was a subspecies of the sage sparrow that was being considered for elevation to its own species. And some phylogenetic work was done. And not only did they elevate it to its own species, they put it in a new genus. So even though California bird life is fairly well known and really well appreciated by birdwatchers and people generally, there's a lot that we actually are just beginning to learn about the relationships of these birds. So this is really a field that is very lively and still requires a lot of attention. So the bell sparrow's abundance, we looked at this one in particular because it was considered an endangered subspecies at some point, or is being considered for endangered status. Fire, the bell sparrow seems to do well with fire, about as well as in control plots, maybe not quite as well as with winter fire. The masticated plots don't attract bell sparrows back. This 0.2 bell sparrow per visit tells me that probably this sparrow is not around long enough to establish a territory. So you might have birds beginning to use plots as vegetation recovers, but you don't see them in the short term setting up their territories again. So that's important to note. Again, we can do guild analysis. Guild analysis is one of the things that methodologically connects the Lyme disease study to this study. And we can look at the distributions of bird plot use by main food item and by the other behaviors that I noted on the last slide. And although these don't look different, the distributions of insectivores to nectarivores, to omnivores to granivores or seed eaters really does change slightly. And you can analyze this using something called the pairwise chi-squared and Fisher's exact tests. And you can see that fire in the fall and fire in the winter basically have fewer insectivores than expected compared to sort of the control plots or the proportions that are represented in control plots. This is kind of interesting just because on the previous slide we see that insectivores are sort of the birds that use this habitat the most. And I can tell you that insectivores are also the ones that tend to migrate back and forth between North and South America when their food disappears in the winter. So chaparral seems to be very important to insectivores. but the fire treatments actually don't attract them back as much. But you do get more granivores than expected. Fire spraying attracts fewer granivores, and mastication, as I mentioned before, seems to attract hummingbirds in the short term. That's kind of interesting. I have a whole stack of results that look like this to do this very sort of fine-scale guild analysis with these birds to see, to get kind of a handle on what's going on with the dynamics of these birds. But guild analysis can't tell you everything. It can't tell you anything about the demography of these birds, like are they trying to nest? How many nesting attempts do they make per season? How good are they at having successful young that fledge out of the nest and survive? This is a kind of interesting thing that just came up out of the data, is that we have all of these birds called flycatchers that are all related. They all have kind of a similar feeding guild, which is called the flycatching guild. They all disappear out of the masticated plots for some reason. I don't know why. It could be competition from other birds. It could be that they don't like the look of a bare plot. It could be lack of perching substrate for them. But we do have a bunch of fly-catching birds that come in. This is the western bluebird. So guild analysis is not going to tell me everything about the dynamics of birds in these plots. I also want to bring up the fact that small gallinaceous walking birds are declining globally. And in Chaparral, people will tell you that, Chaparral is full of quail. So you have a California quail on the left. You have a mountain quail, which is it's a bird that's incredibly hard to see. That if you're a birder, this is a really, it's very secretive. You're really lucky to get a chance to see one of these birds. But around the globe, you see over and over and over again, quail and small partridges are just declining in number like crazy. They're some of the most endangered birds, and they've decreased at an incredibly rapid rate. Most of the time, the story for these birds is that they live near agricultural regions, and the intensification of agriculture is removing scrublands that they need for cover, they need for forage. I'm going to just throw this out there that this may be going on in California as well, but nobody's studying this. Birds like this need very clear understories to walk around and shrub cover that protects them for predators and protects their young from predators. Their young are flightless and they walk along the ground. These birds are excluded by grasses. So once you have tall grasses, they can't move around anymore. It's too energetically costly for them to move around. So connecting the rise of annual grasses from mastication to the sort of the feeding guild, the morphology, and the energetic constraints of small gallinaceous walking birds, this is really key to focus in on. And this is a reason for hunters to get involved. They may be concerned about the persistence of quail in California, and birders to get involved, because birders love these birds. They're very pretty. They're very charismatic. So this is something to watch in Chaparral as Chaparral is changing. And the final thing I'll say about this is that these birds are very heavy. They walk a lot of places, and they're not quite as agile as other birds, meaning they don't move around quite as much. A recent study by Liz Van Mantjam, Marty Witter, and John Keeley have basically shown that animals either have to persist through fires or they have to flee and recolonize areas. And this is a case where the increasing size of fires in Chaparral may be imperiling these birds as well. And also, if as we know, from studies in the Moritz lab, let me see if I can pull this up here. Yeah, by Philip Dennison, Max Moritz, and others, a study called Large Wildfire Trends in the Western United States, 1984 to 2011. The size of fires in Chaparral is getting larger. What this means, according to Liz Van Mancham, Marty Witter, and John Keeley, is basically that the birds are not going to be able to recolonize burn sites from these affected metapopulations. So the metapopulations of these birds are going to be greatly affected by fire sizes, which we expect to keep increasing. Okay, so synthesis. I have presented today 3 mechanisms by which Lyme borreliosis becomes more prevalent in California wildlife with chaparral removal. These are all testable. I wish I had more data. I can't say that this is going on for certain, but this is an area that I hope to attract scientists to. Chaparral-associated birds have fewer ticks compared to birds primarily associated with other habitats. Competent carriers of BBSS, the spirochete that causes Lyme disease primarily, are primarily not chaparral birds. And the competent carriers disproportionately pass that bacteria back to ticks. Finally, competent wildlife hosts of BBSS, such as American robins and dark-eyed juncos, replace the non-competent hosts in masticated areas of chaparral. Synthesis 2. Land use is tied to emerging infected diseases. This is not contentious. Fire management and fire management choices in California are also going to affect disease ecology. I've shown that fire management in chaparral is detrimental to bird abundances generally and to a large number of species, especially the rare birds. Mastication is more harmful than fire as a management tool. Fire management choices may be influencing the incident rate of Lyme disease in California, and fire managers should not overlook disease ecology consequences. And on this last point, we don't have a lot of information about disease ecology in California. So my management recommendations is that given that chaparral biodiversity is already threatened by mastication, too frequent fire, and habitat fragmentation, which has been shown over and over and over again in different studies, including the ones I have here, we probably should limit management activities to what is essential for the protection of human health and well-being. The management activities that we're doing are just beginning to be known in terms of Lyme disease ecology and disease ecology in general. Okay, I think that is it for me. Yep, that's it for me. So again, thank you for joining me. I'll take questions if you have them.
Okay, so first I want to give a giant thank you for Erica. I know I learned so much from that in the last few minutes. So now we do have a few minutes left for some Q&A. The first question was for me, which is the link for these slides. So if you look at the web links page down here, the second link down, the CFSC webinar page, that will have the link to the PDF of these slides. It will have the recorded link once I get it edited, as well as a list of links that Erica has provided me. So it's links to the journal article she mentioned that was published and some other resources that you might be interested in. So I would definitely recommend, I'll put some of those links up today, and then I'll probably get the recording in the next few days. So that will be on that page. And with that, so if you do have a question, please type it in your chat box there. There's a few other links that we put up, or Erica, do you want to explain the other 2 links that are under the web links there?
Oh, sure. Sure. So my analyses relied heavily on categorizing birds by their behaviors. So I actually used the categories provided by the Cornell Lab of Ornithology. So that first link is range maps, songs and life history information about North American birds, with the Lawrence's goldfinch as an example. But you can basically navigate through all of the all of the birds that are present in the United States through that website. It's really a wonderful resource. Also, I have to say that after I give talks like this, I often get approached by people who have Lyme disease or who have a loved one who has Lyme disease. And I can tell you just immediately that I am not an expert in the literature related to Lyme disease in humans. So I'm putting up a link from the Centers for Disease Control and Prevention that itself has resources telling you about the state of the field, the state of what's known for human treatment, and range maps for the various outbreaks of Lyme disease.
Great, and I'll point out the last link we didn't talk about, which if you've been with us before, if you've heard this plug before, which is the webinar evaluation. This is a 10 survey questionnaire that's just online. It takes a couple minutes to fill out, but the responses really do matter, both if you give us specific feedback about how we can improve our webinars in the future or topics you'd like to see in the future, but also allows us to report back to our funding organizations that this is still a successful tool to be using to disseminate science information. So I do highly encourage you to click on that link before, or to either copy that link, or I'm also going to send with that email in the next day here a link to the evaluation, as well as the link to the recording and all the links to the page that Erica has provided for us. So we've got a couple minutes left. I think, any questions there? Someone's typing, but it might just be a thank you. So just a comment from their personal experience that CDC is still in denial about the severity of Lyme disease. And she provided another source, which is lymedisease.org.
That's a great thing to point out, that the CDC has a lot of information, but there's a lot of other information out there. Some of it, some information on the web about Lyme disease is kind of predatory because it is actually taking advantage of people who really need help and are very, very much suffering with the disease. So I'm going to give another shout out to this link, lymedisease.org. That's a great source of information. And I will say about the state of Lyme disease research that the field itself is quite old. The people working in this field are using techniques that could be updated. There is a lot of disagreement in the field about how many species of Borrelia there are and whether they cause different diseases. So just as sort of an informational point, scientists are aware that that there is kind of disagreement in the field. But I will say that whether or not you agree with the assessment done by the CDC, Lyme disease is, like I said, globally distributed around the temperate part of the world. And so we have people working in Europe on this problem, people working on Asia in this problem, and we're going to be seeing a lot of new information and a lot of new techniques coming up rapidly. And that's very exciting to me because I grew up in an area where people contracted Lyme disease at a really high rate. I would love to see this solved for people.
Well, and it looks like we do have one question, which is a question I think all researchers love to hear, which is, what is the next study that is needed to understand this public health concern?
Oh my goodness. Wow. Okay, the next study. Okay, so I did a study that just sort of, I did two studies that coincidentally shared Chaparral as an overlapping ecosystem that that basically was pretty interesting for a number of reasons. But if you talk to hikers in California, they will tell you that the real tick populations are out in the coastal scrub. And coastal scrub is the other ecosystem that's most heavily impacted by development. So, so first of all, I think we need an education program for doctors, telling them that Lyme disease is in California. But secondly, we need more ecologists working on this problem in other habitats, including coastal scrub. And if you think about it, I just talked about 2 studies that people had to write grants for, to get money for. They were long-term studies. They took many years. They required a lot of scientists to work together from multiple disciplines to start establishing what's going on. I would say the next thing that could help us in terms of preventing preventing unwanted consequences like disease ecology, things that might be coming down the road that we're not even aware of yet. We probably should be as fire scientists, as land ecologists, we should be asking for public lands to halt management activities in Chaparral. And we should be asking for federal level protections for Chaparral so that it's not removed and biodiversity is not coming into contact with human settlements in a way that is leading to the outbreak of Lyme disease and possibly other things like Borrelia bizzettii and so on. There are forest inventory systems run by the USDA. The Army Corps of Engineers has a program to protect wetlands over 1/4 of an acre. I don't see a reason why Chaparral can't come into a Chaparral inventory system where there needs to be a particular reason to manage it rather than management being the default action. The science is pretty clear that management actions lower biodiversity. So I would say that those are the next directions for this kind of work.
Sounds like we have a lot to do. So with that, we are out of time, but I again want to thank everybody who joined us and Erica so much for a great presentation, and you'll be hearing from you shortly with that email. So thanks again and have a great rest of your Wednesday. Thank you.
Yeah, take care. Bye.